Well, hello everybody. I'm excited to talk about DIYgenomics, do-it-yourself genomics. This is an open platform for citizen scientists to organize your own studies, linking genomic data with phenotypic outcomes, and to build personal genomics applications. DIYgenomics is a nonprofit organization that was started in March. We have 15 team members currently. And my own background is primarily in innovating and implementing science and technology. The structure of the talk is to first provide the context for DIYgenomics, and then the motivation, and finally our specific activities. So we're familiar with open source software in the literal sense, but conceptually open source is starting to permeate more aspects of society. For example, in the data and data analysis area, what if we had million-member biobanks with user-contributed data? So my doctor can't open source my genome file, but in fact, I did that myself on the web to the SNPpedia 2 years ago. Another aspect is activities. So science in particular is starting to see open source approaches of collaboration between scientists and starting to look at problems that are uninteresting to traditional researchers or pharmaceutical companies, for example, to do the long tail of medicine, where numerous individuals are afflicted by conditions, but nobody else is looking into them yet. The ecosystem of science has, of course, traditional science, and of which a small number of people are doing open science. This means essentially sharing data in usable formats, collaborating on hypothesis formation, and publishing failed experimental results. In parallel, there are many citizen science organizations ranging in scope of activities from, for example, Galaxy Zoo for astronomical data identification and annotation, to eBirds.org, to the burgeoning DIY bio movement, which has been on the scene for the last few years, and of which DIYgenomics is one flavor. So why genomics? Why now? Traditional Moore's Law improvements run at a rate of 1.5x per year, but genomic sequencing has been running at an improvement rate of 10 times per year, as indicated in this logarithmic chart of the number of base pairs sequenced per dollar for the last 30 years, with a significant uptick in the last several years. I'd like to suggest to you that this rate of progress may continue because we're just on the cusp of third-generation sequencing technologies. In the old days, i.e. 10 years ago, when the Human Genome Project was in process, the state-of-the-art technology was first-generation sequencing, Sanger sequencing technology. This technology consists of making millions of copies of different sized chunks of DNA strands, printing them all out on an electrophoretic gel in size, grouped from largest to smallest, and reading out the base signature. The second generation of technology, which is the current version, consists of parallelized sequencing. This method takes millions of single-stranded copies of DNA and puts them on a microfluidic chip, then floods the chip with a solution with the different base pairs, and as the base pairs attach to their complementary strand, read the signature as a fluorescent label is given off. And the third generation of technology is just coming into play now. The leading company is Pacific Biosciences, and they claim a 30,000-fold improvement over current methods. And the industry seems to agree. They gobbled up 10 of their beta machines at $800,000 a pop earlier this year. This technique takes advantage of the natural process of the DNA polymerase enzymes copying of DNA strands, here looking somewhat like Pac-Man as it copies a DNA strand. Again, the bases are read as a fluorescent label detaches during the copying process. The fourth generation of sequencing technology is also already under development with companies like Oxford Nanopore, Ion Torrent, and Halcyon Molecular. This advanced technology just reads the electronic signature of the base directly as it's pushed through a nanopore. Not only have we been making progress in reading DNA, but also in writing it. We'd like to synthesize it. A landmark announcement was achieved in May of this year when Craig Venter's lab announced that they had printed for the first time a synthetic chromosome, put it into a cell, and gotten that cell to boot to life. At the same time, our notion of health and health management is changing. For example, along the x-axis, we appreciate that we'd like to include different kinds of data streams into identifying an individual's well-being and health, and together with an upstreaming of outcomes on the y-axis from not just cure, but also starting to look at prevention, enhancement, and so on. The individual is the center for action taking, possibly in collaboration with peers and professionals more distantly. Our activities at DIYgenomics consist of looking at this problem of the lack of predictive health management tools. We have some solutions to this. First is our crowdsourced research studies, where we're attempting to establish the baselines of wellness and preventive interventions. Second, we have a suite of personal genome analysis apps that are open source software. And third, through our partner company, Genomira, we're creating a platform for citizen science where users can easily share their 23andMe or other data and also post up a study like the eBay of genomics. Post up the research you would like to do and attract participants. Our fun personal genome apps consist of an Android app, which has almost 1,500 downloads. It's free and provides a side-by-side comparison of the top 20 health conditions covered by consumer genomic companies, the specific variants that they look at, and their research references. The same thing is available on the iPhone and on the web. Here you may notice in the top center an option to privately view your own data. Of course, we're very conscious of the fact that many people are quite concerned about the privacy of their own genomic data. So we created a Firefox extension, open source, where the user may, in these 3 steps, upload their personal data as a Dojo widget overlay to the logic of the website. We also have a drug response application where users may investigate for 250 different drugs, their particular response or side effects, and we'll be launching an athletic performance evaluation app soon. The other arm of our activities is citizen science research, where, again, we're looking specifically at preventive measurement, or preventive medicine, establishing baselines of wellness measurement and preventive interventions. I don't want to show up with diabetes being diagnosed in 20 years. I like to start watching my markers predictively now and intervene so that I don't get sick later. We're integrating the available health data streams of genotypic data, phenotypic data, and whatever else we may be able to in a series of studies that we've outlined on our wiki in both health genomics and behavioral genomics. Our first study is in process now, looking at 2 mutations in the MTHFR gene that may impact 50% of the population and lead to a possibility that vitamin B9 is not converted to the active form and absorbed by the body. Here is the biological pathway of the phenomenon. The ones indicate the places where traditional multivitamins impact the pathway, and the number 2, where the active form of B9 is required. As usual, in our study structure, we identify the genotypic markers, then the specific physical biomarkers that we'd like to measure, and the appropriate corresponding interventions. Here, each subject acts as their own control by doing all 3 interventions in a serial fashion. Here, for the first time, are our citizen science results of our genomic study. We have 5 participants in our pilot cohort, and realize this is not statistically high-powered, but we are looking for an example of an effect. What we see on the left is our genotypic data with the risk mutation in red, and on the right, the values from our homocysteine blood tests. The first test was a baseline for all 5 of us. The second, after 2 weeks of taking a spectrum multivitamin from the drugstore. And the third test, after the intervention of taking the active form of B9, L-methylfolate, and you can see the results that 4 out of 5 of us decreased our homocysteine levels, which is good, just by taking the Spectrum multivitamin. And then some people improved further and some didn't by taking the B9 L-methylfolate. So it's a little early to conclude finally from this study, but an example of the citizen science that's going on, and you're free to follow our results at that wiki link below. It's clear that the amount of biodata starting to be generated on each individual is growing, and that there will be huge demand for quantitative self-tracking tools on the order of establishing a whole new industry. And we're innovating the research model for this. In the traditional research model, you have in the center an institutionally based principal investigator that conducts studies on research subjects under the purview of an institutional review board, supported by grant funding and publishing their results in journals. In the citizen science model, things are a little bit different. There's no distinction between the principal investigator and the research subject. All investigators are also subjects or participants. Therefore, the analog to the institutional review board may look a little bit different, and it may constitute citizen ethicists that are, for example, mentored by other professionals who have done this before, or a list of FAQs. The funding for citizen science could come from patient advocacy groups, research foundations, social venture capital, and crowdsourcing, and the results could be self-published on the web, inviting commentary. The second study that we have in mind is more complicated. It deals with aging. First, we'd like to check gene expression for 10,000 individuals in the peer cohort. We'd like to compare the physiological age versus the biological age. Then we'd like to do our usual, identify the top 10 genomic biomarkers of aging, their corresponding phenotypic, the physical measurement of aging, and try applying corresponding interventions in a longitudinal study with ongoing measurement. Please join us by participating in a study, participating in our open source software development, organizing a study on this platform, contributing your data, or making a donation. Thank you. And the slides are on the conference website for this session. Any questions?
So if sequencing is getting so cheap, is the 23andMe SNP data really even going to be that useful? I was thinking, why not just get a full sequence?
Yeah, exactly. So in a 2-year time frame, almost, it seems like why bother with the $400 23andMe now? Because maybe whole human genome, which is currently $20,000 for retail customers, maybe is of a similar price. And in fact, we would assume that companies like 23andMe may start offering whole human genome as an offering as it becomes available. So it depends. I usually tell people if there's a health issue you really want to look into, go ahead and get your 23andMe data now. But if you are price conscious and want to wait and don't want to participate in citizen science and so on, no reason to rush into it now. Things are getting cheaper all the time. Back corner.
Well, I have 2 questions. First of all, thank you. Really interesting stuff. Thank you. And I have 2 questions. One was sort of more general, and the other was specifically about that study.
Yeah.
So the more general one is, if you're crowdsourcing your research subjects, how do you deal with selection bias?
Mm-hmm.
And the more study-specific one was, were both treatments done in the same order for all subjects, or were there washout crossover periods and so on like that?
Yes.
There could be an order effect, and also really you usually have a washout period between treatments.
Mm-hmm. Mm-hmm. Yeah. So the first question. So yes, we realize we have selection bias that people who have ordered 23andMe customers, of whom there have been publicly announced to be over 60,000, is already a self-selecting group. And in our study, also, we have some international collaborations. So we have a peer cohort in Japan that we're trying to replicate these study findings in. But our own cohort is very homogeneous with these 5 people. And so we realize that there's a barrier to that But as we expand the research into the 10,000 person aging cohort, that'll be much more heterogeneous. In regard to the interventions and the cycle and so on, so we did check with some homocysteine experts, essentially, who counseled us on the order that we're doing in the study. And instead of doing, we'll be doing another washout at the end, but instead of doing a washout each time, it was felt that you'll see an effect after only 2 days, and that the specific process we're doing, which is the multivitamin followed by the L-methylfolate alone, followed by both together, is fine to eliminate any effect and not really need a washout period in between.
Okay, thanks.
Mm-hmm. There was another question back.
Yeah, I mean, so your talk rocked my world. I thought this kind of possibility to empower patients was decades away, and you're telling me it's starting now and years away, which is great news.
Mm-hmm.
The question I have is, I have felt like as far as open source healthcare software goes, and it's non-genomic, that we don't have our act together well enough to support what you want to do as far as your phenotype data.
Mm-hmm.
I was just wondering if you could comment on that problem, or how you think about that, because you're obviously to the point where you need it.
Yeah, exactly. So we're inputting our PDF blood test report data into a wiki at the moment, so it's not very automated. But what we see doing is similar to the Harvard Personal Genome Project, which is using Google Health as the phenotypic data collector, and then just linking that to permissioning in access to Google Health from the Genomira platform for our phenotypic data collection.
May I suggest that an in vivo X module for what you're doing is probably relatively obvious.
Mm-hmm.
And would give you vastly greater depth of data and more control.
Mm-hmm.
More importantly, you could set up an instance for yourself.
Mm-hmm. Great.
And so I'd encourage you to look at that if you're interested in PHRs. Is the phenotype
Mm-hmm.
access.
It would be nice for us to show leadership on that as well, and particularly including the other dimensions of phenotypic data that we'll be starting to collect, such as telomere length and other very specific factors for our studies. I appreciate their recommendation.
So the study you did, who collected the data, who did the measurements? Was that the individual as well?
All of us. So it's blood data. So we ordered on the internet blood tests, and they were performed at local LabCorp facilities. So that's essentially how they were generated.
Going on from this,
Mm-hmm.
how would you cope with kind of bias problems with more subjective phenotypes? So
Mm-hmm.
looking at the effect of pain relief medication.
Mm-hmm.
It's not really something that can be done at the individual level. You can't do a blind study that way.
Mm-hmm.
How do you handle that?
Yeah, so definitely we want to incorporate patient-collected data as an attribute, and in fact we have a collaborator at UCSF in the cancer department who is enlisting our help on that kind of a study. So patient-collected data is seen. So essentially, how you avoid some of the bias and other data collection issues are just statistically. So with the numbers, then things tend to even out into quality data. And further, that non-interventions that are just, say, a daily diary, or you're notified to check into a mobile app with your current level of pain or so on, are seen as a much more user-friendly version of community science that is actually quite a hassle for people to go over to LabCorp and get a fast overnight and go over there and get a test and so on. So we'll definitely be starting to include that.
Did you ever go around looking at open source tools for the more traditional side?
The genomic.
Citizen approach.
Oh, I see.
The traditional approach.
More open software tools?
Yeah, if you were doing like a blind study, you're doing a blind study, you're using genomic tools on the open source side.
Yes, I guess we haven't done a lot in that traditional format. I'm not sure if we could apply. There are many open data sets that we use all the time in the genomic databases, for example, but we haven't really had a software. So that's more what our partner company is looking at as they build out the citizen science platform.
Just out of curiosity, on your aging study, what are you looking at in
Oh yeah, definitely. We have several. So there are, and I'd be happy to send it to you later. So there are many things for which there are interventions. So the classic case is Alzheimer's, that there's no clinical treatment currently. However, as of 2 years ago, we've known that about the correlation between midlife cholesterol levels and Alzheimer's onset. So that's already something that people can start getting active with, and that the Boston University REVEAL study indicated that 70% of people finding out that they were ApoE4 positive, which is the risk allele, in fact, did change their behavior in some way. Some of the other things, so we're looking at, there are several things, for example, RNA editing mutations may be, one of the interventions may be speeding up metabolism so there's quicker turnover in cells. So there, just as an example of things that we obviously don't have a gene-based therapy for yet, but there could be some sort of intervention that you might try if you know you have a lot of mutations for a certain condition. Telomere length, we don't have a good solution for that yet. Lots of people are looking at that. Anybody else?
My wife's a genetic counselor, and so she deals with the human side of this and the testing. And we're getting to a point where people are going to be able to find out a lot of genetic information and consequences of this. Do you have anything that's addressing, the interventions that need to take? Are they appropriate? The counseling that needs to be done, because there's a lot of psychological issues. What are your plans?
Yeah, definitely, and I think genomic data interpretation on the personal level is one of the biggest opportunities and challenges we have generally in the medical field currently. And so what we are doing specifically is we assume people have already sort of sorted through a lot of those issues before they arrive to us and refer out to genomic counselors and so on. But if you have your data, if you've already received your data from a service like DIYgenomics or 23andMe or Pathway, or et cetera, then you may have already had access to their genomic counseling function. One thing that has come up for us that's pretty interesting is in terms of data sharing. So we have several individuals that have contributed their whole 23andMe data files to us to help with our study structuring and investigation of genotype prevalence in our peer community and so on. And so it's been quite interesting. Some people, quite eager to share their whole thing. They're radical transparency advocates. They think the benefits outweigh the costs. And other people, don't feel that way at all and want to, only, okay, sure, when you need these SNPs, I'll tell you my data. And then an interesting aspect that's come up is the consent process. So we do ask people to discuss it with their family because exactly because some of these issues. What are the ethical norms around your own genetic data? We think it's ours, but in fact, we share a lot of it with family members. And so one interesting potential standard that happened was one participant did a video consent of his family members, his kids, assenting to him contributing his data. So I thought that was sort of interesting. Anybody else? Great, thank you.